Yeah, if you get that, thank you. I appreciate it. All right, guys, so our recording is rolling here, so let's get started. Morning. First of all, a quick review of what we've already talked about. Nothingness. So we talked about two different parts of your immune system. The non-specific, or sometimes it's called innate part. And we just blanked out again. And also the acquired, or sometimes it's called the specific part of your immune response. Now, while this is up, remember, non-specific immunity protects you against things in general. You come born with this. This is not something you have to develop or get over time. The skin's the most important part. It's a barrier. It makes secretions to kill germs. Inflammation, remember, is a good thing, at least in moderation. It happens when there's damage to tissues, burns, cuts, whatever it may be. Cells in those damaged tissues called mast cells essentially give off SOS signals, either called histamine or prostaglandins. And those tell the blood vessels in the area to do what? Send more blood, dilate, send more blood, send me more platelets to clot this mess, send me more white blood cells to kill the germs that are in me. We talked about interferon being a protein we make to interfere with what? The lytic cycle, the virus reproduction cycle. We talked about your stomach acid kills things that come in through your mouth, denatures the proteins that they're made out of. We talked about the normal flora being the bacteria that normally live in and on you. And we talked about how they sort of act as almost like an offensive line. They block out things that would grow otherwise. And that's why sometimes when you take antibiotics, you get what are called secondary infections. Because do the antibiotics just kill the thing you're trying to kill? No, they kill some of these. And therefore some germs that might not be able to grow instead. The phagocytic cells in your body, like the macrophages. And you've got this other kind of cell called the neutrophil. Those are also non-specific. They kill anything that's not supposed to be in you. They're not looking for specific invaders most of the time. We mentioned how that in your saliva, there's this stuff called lysozyme. It's also in tears, and it helps to kill germs. The mucous membranes help to filter. We said they also help to do what other two important things to the air that we're breathing. Humidify it, give it a little moisture, and also warm it up if you're in cold places. And we talked about pyrogens, being proteins that help to raise your temperature, cause a fever, and that helps your immune response to work better. But all that stuff's non-specific. The specific stuff is what we want to focus on today, and a couple of autoimmune things as well, if our computer will cooperate. So we'll start back with our picture that you guys should have memorized for sure, as many times as you look at it. Remember, there's two parts to your specific immune response. The picture on the left, on your right. What kind of immune response is it about to start? This picture with the purple cell, what part of the specific immune response is it about to trigger? Remember, there's two parts. Cell-mediated immunity and humoral, also known as antibody-mediated. That's what this macrophage is about to start, the humoral, or the antibody-mediated. So it phagocytizes the germ, puts it in a little vesicle, uses enzymes from what organelle? Lysosome. It does intracellular digestion on it. It breaks it down into little pieces. What do we call those little pieces? Antigens, or proteins, which it then is going to put up on this MHC class 2 protein that's found on your phagocytic cells. And what do we call the act of holding them out there? Antigen presentation. It presents those antigens to what kind of cells? Helper T cells. And they physically have to dock together. And the helper T cell, what's it learning about that antigen? Its shape. Remember, only helper T cells that have a receptor that can fit to that antigen can bind. Once they do fit, they become activated. What do these helper T cells then do? Yeah, they start sending out signals. And we said they sort of start acting like Paul Revere. All right, so far we've looked at this middle part of the picture. Right in there. Yeah, it's a little behind. The helper T cell is going to send out chemical signals next. And those are sometimes called interleukins. Go ahead.
So the B cell and then the those are called interleukins?
Yeah, there's different kinds of interleukins. Yes.
But just like whatever the helper T cell stimulates is an interleukin.
The signal that it's sending out is an interleukin. Yeah, this is a name for a signal. It means a signal between, leuko means white, like white blood cell. But remember, that signal is only going to stimulate B cells that are wet. It's only going to stimulate B cells that are specific to what? That antigen. And what's the first thing it tells those specific B cells to do? Copy, clone yourself, because there's not very many of you. Make copies. And then most of those cloned B cells become plasma cells. They metamorphosize into plasma cells, which start making what? What do plasma cells make? Antibodies, which attach to that specific antigen, and in one way or another, cause it to be gotten rid of, right? They may signal macrophages to come eat it and kill it. They may act like little handles for other white blood cells to take that germ and help to flush it out your urinary system. But in one way or another, they help to get rid of it. After a few days, what happens to the plasma cells? They die. What's the word? Apoptosis. But what doesn't die are memory cells. Cells that never became plasma cells, but cells that know what this particular germ looks like. And if you ever get it again, you skip most of those early steps of the process, and you make so many antibodies so fast that what never happens? You don't get sick. You don't get symptoms. That's antibody-mediated or humoral immunity. And remember, that only helps you if the germ is where? In fluids, because antibodies are what? What are antibodies? They are proteins, which are big and bulky, and they can't get into cells. So we'll come back to this and talk about the other part in a second. Let's go to a picture here first. Something to remember. We only have a few genes that code for antibodies. A handful. We talked about last time that in your DNA, what's the part of the DNA that actually codes for something called? We've got parts of DNA that seem to be coding and parts that don't seem to be coding. What are the coding parts called? Maybe not. There we go. All right, somebody said. So let's say this was a sequence of, let's just say this is messenger RNA. This is mRNA. So it's just been made from the DNA. We have these pieces of DNA that are called introns. And for a long time, scientists thought those were just junk. And before the proteins were made from this mRNA, what happens to those intron sequences? They're cut out. So let's say that this right here is an intron. This gets cut out. And then this piece, and this piece, and this piece tell your ribosomes what protein to make. But what scientists have learned recently is that, yeah, sometimes this piece right here may be treated as an intron. But if something different is going on in your body, maybe this piece is treated as an intron. And this piece is coding. What that means is each gene is actually what? Can code for lots of different things. So one gene is really lots of genes poked into one basket. And that idea is called alternative DNA splicing. What that means, essentially, is that even though we only have a few a few genes that code for antibodies. Now we can end up with more than 1 million different kinds because these few genes can be spliced together to make lots of different proteins. When we get to genetics, we'll talk a lot more about that. Let me go to a picture here of an antibody, first thing. So here's an antibody. If you've been in class the last couple of days, we've been talking about enzymes and active sites and stuff. And something that I want you to mention or notice, that looks a lot like an active site, right? And we keep saying with enzymes and active sites, substrates and active sites, they have to fit together how? Like a lock and key. The same goes for an antibody and a what? Antigen. They have to fit. And that's why antibodies are specific. So this antibody, this whole thing is a big old protein. The part that says C on it is constant. It's the same in every kind of antibody. But the part up here that says V, that part is different. And that's what allows different antibodies to attach to different antigens and hopefully help us to get rid of them. So like we said before with antibodies are proteins made to attach to specific antigens. They can act like tongs that let white blood cells use them to grab onto and transport pathogens, hopefully to the urinary system so we can flush them out. Remember that piece, that antigen bonding site that we looked at, is sort of like an active site on an enzyme. And it's different for every antibody type. The constant region is the same in every antibody type, and it acts almost like a handle for the white blood cells to use. And then also remember that even though we only have a few genes for antibodies, that because those few genes can go through this alternative DNA splicing setup, those few genes can actually code for lots and lots and lots of specific different antibodies. All right, so let's go back and talk about the other side of the picture here. We looked at the picture on the left a lot too. We said that orangey yellow picture represents what? A somatic cell. All your cells have an MHC called the MHC class 1. And it is supposed to present just proteins from inside the cell, which normally should be what kind of proteins? Normal, healthy proteins. And if it's presenting normal, healthy proteins, it's sending what message to your immune system? Leave me alone, I'm good. But if something weird is going on inside, the virus, cancer, some of the proteins that get presented are probably dangerous ones, deadly ones. And in that case, let's go back to this picture. It might present those antigens to a helper T cell, just like we had here, which would stimulate cytotoxic T cells. Those antigens might be presented directly to the cytotoxic T cell. Either way, we've been calling those not cytotoxic, we've been calling them what? Killer T cells. Once that killer T cell is activated, it goes out and starts looking at the MHC1s of your somatic cells. And it's looking for cells that are displaying a specific antigen. And if they're displaying that antigen, what's it do to them? It pokes holes in them, it perforates them, kills them so that they don't spread that infection. And just like before, a lot of these cytotoxic T cells, first of all, they clone. What happens to most of the active cytotoxic T cells after the infection's over? They die. But you generate memory cells at this place too, this particular junction. So these two things work together, and they make up your specific immune response. The left part makes antibodies, which kills germs that are in your body's fluids and in between the cells. The right part kills your own cells that are sick so that they don't spread. And that part is called cell-mediated immunity. But together, this whole picture is your specific or your acquired, because you have to build it up, immune response. All right, a couple things that I want to touch on that we haven't spent much time on yet. Let me get to where I want to go here. So we talked about different kinds of immunity last time. And we said that natural active immunity is your normal immune response. You get sick, you make antibodies, and you make memory cells, so that the next time you get sick, or the next time you get infected, you don't get what? Sick. That's natural active immunity. It's the normal way that we work. We said that natural passive immunity is when you get antibodies from who? From your mom. And that can happen across the placenta while you're still in the uterus, or it can happen how? Through breast milk. We talked about artificial active immunity, and that's an example of how what works. Vaccinations. When they give you a vaccination, they're either giving you pieces, like antigens from a particular germ, or they may give you what's called an attenuated version of the germ. For example, when they give you a flu shot, a lot of flu shots have active virus in them. But they're human viruses that instead of being grown in humans, they've been grown in what? How do they create the flu vaccine? They grow it in eggs. So as it's growing in the eggs, it evolves to infect what? If you're growing in an egg, what do you want to evolve to infect? Chickens. It's chicken eggs. They grow in chicken eggs. So they evolve to infect chickens and not humans. And they do that for several weeks. So that by the time they take it out, it's different than the human virus that would make you sick, but it's still enough like the human virus that your antibodies will what? Will attach to it and kill it. So it shouldn't make you sick, but it should still give you memory cells and antibodies that will protect you from the real virus. Sometimes we say attenuated is just weakened. It's not so much that it's weakened though, it's that it's been grown in a different thing, usually eggs. And it's evolved to infect egg cells, chicken egg cells, not so much human cells. And then artificial passive immunity, we talked about, they used this in the hot zone. They took blood transfusions from people who had what? Survived Ebola. And they gave those to people who were still sick in the hopes that the memory cells or the antibodies in that blood would protect them. All right, and another key thing to understand is this diagram here. This is set up to be about vaccinations, but let's ignore the vaccination part. Let's just say it's about when you get sick. Right here, you get a germ for the first time you've ever had that specific germ. And your immune response is kind of slow. It takes it 5-6 days to get kicked in. But slowly you start making antibodies, and the amount of antibodies in your blood starts to go up, and eventually it tops out. And by the time it tops out, what can we probably say about you? If your antibody levels have topped out, what does that probably mean? You're better. So what starts happening to the plasma cells that were making those antibodies? They start dying, and the antibody levels start to drop. And they drop back down to almost zero. But what's still in your blood? Memory cells. This first time you get sick, your immune response is called the primary response. We use primary to mean first. And your primary response is sort of slow, and it's not very big. But right in here, about day 60 or so, let's say the same exact germ gets into you again. You've already fought it off. You already know how to fight it. You already have memory cells. So almost immediately, within just hours, you start making antibodies. And this time you make the antibodies way faster, and you would make way more of them. That's called your secondary immune response. And it's so much faster and so much bigger that what never happens. you don't get symptoms. So know the difference between a primary and a secondary immune response. All right, and then winding it up, we'll talk just a little bit about autoimmune diseases. Autoimmune diseases is when your own immune system starts to attack your own cells, your own healthy cells. We said that in most cases, who's affected by them? Who usually gets autoimmune diseases? Almost always women. And lots of times it's women who've had a child. Sometimes that can confuse your immune system. But there are exceptions to that. There are men who get these from time to time. One of the most common ones is lupus. And this is one of the few of these that is systemic. What do we mean by systemic? If we say a disease is systemic, what do we mean? It causes symptoms not just in one place, but all over you. And because it's systemic, it's really hard for doctors to diagnose. It affects different people different ways. Sometimes it just causes a rash. Sometimes it attacks your kidneys. Sometimes it attacks your joints. A lot of times, if you ever watch shows like House, when they can't diagnose what the problem is, they'll say, she's probably got lupus. Because they just don't know. It's hard to diagnose. Rheumatoid arthritis is, you see a lot of commercials about this on TV for drugs to treat it. This is when your immune system starts attacking the cartilage in your joints. And it causes your joints, especially in your fingers, sometimes in your knees, to get all crooked and bent and twisted. It's very painful. It's debilitating. If you have rheumatoid arthritis, it's going to be hard for you to work because it just hurts to move. Type 1 diabetes is actually an autoimmune disease. When do people typically get type 1 diabetes? Sometimes it's called child onset diabetes. And this is when the immune system attacks the part of your pancreas that makes insulin. That part's called the islets of Langerhans. So you lose your ability to actually make the insulin. Your immune system kills the parts of your pancreas that makes it. And then we also have multiple sclerosis, which is when your immune system attacks the myelin sheaths. These are like the insulating parts around the axons of your nerves. If you took a wire and you removed the insulation and you surrounded that wire with the water, what would happen? You have an electrical wire running through water with no insulation. Bad things, right? It's going to short out. And that's sort of what happens with your nerves. These myelin sheaths, this insulating part of the axon is removed, and therefore the messages don't get to where the messages are supposed to go. And you lose control of certain parts of your body. Again, mostly women are affected by MS, multiple sclerosis. These are all examples of autoimmune diseases. There's a whole lot more. Some of them are really, really strange. And again, sometimes really hard to diagnose. I'll come back and we'll see what's on the next slide here real quick. All right. So if you're still writing, I'll give you a minute to do that, and we'll look at the last slide, and we'll be done for today. All right, the last topic we will mention is what are called immunodeficiency disorders. And this is when your immune system is just not working properly. The most famous one of these is HIV. HIV causes AIDS. What does AIDS actually stand for? It's an acronym. Yeah, it stands for acquired, which means you have to get it. Immunodeficiency. Syndrome. Acquired immunodeficiency syndrome. We know it's viral. When did it show up in the U.S.? When did we first realize what AIDS was? In the 80s. That's when I was a kid. Early 80s. It's kind of scary at that point in time. But largely in the United States, I won't say it's under control, but we've sort of figured out how to deal with it. We know how it's transmitted through body fluids. We know how to screen blood against it. That was a big problem in the beginning. Who were one of the biggest groups of people that got AIDS in the beginning were hemophiliacs, people with hemophilia. How did they get it? Blood transfusions. Because they were always having to take blood transfusions, and we didn't know how to screen the blood for HIV. So these people went in to get a blood transfusion, and yeah, they got the blood transfusion, but they got HIV to go with it. We now know how to screen the blood. We can protect those people. We also now have antiviral drugs that may not cure it, but at least they stop its progression. People like Magic Johnson have been on those antiviral drugs for 20-something years, and he's still going strong. So in the U.S., we've sort of conquered it, not completely. Where's the biggest problem at? In Africa. One of the biggest killers of people in Africa right now is AIDS, largely because they can't afford the drugs to slow it down. And the education of how it's transmitted is not always there either. The last one I'll mention for today is this one called SCIDS. That's an acronym too, and it stands for Severe Combined Immunodeficiency Syndrome. This is genetic. This is essentially when you're born without a working specific immune system. You guys have probably heard of a case of this before. There was a famous movie made back when I was a kid. It's called The Boy in the Plastic Bubble. John Travolta was the star of the movie. It's based on a true story. This kid was born essentially without an immune system. And they put him in this filtered bubble. He lived his whole life. They turned his bedroom into this filtered, enclosed room. All the air coming in was filtered. All his food was sterilized. If somebody wanted to touch him, they had to use gloves and reach through the wall essentially to hug him. He lived to be up in his teenage years. Decided at one point in time he wanted to go to school. By this point in time he was a celebrity because this is a very rare thing. He was on People magazine, stuff like that. How did he end up going to school? NASA heard about him, and they donated an astronaut suit to him. So he went to school in an astronaut suit. Because all the air and stuff is, it's an enclosed system, so it was filtered and it was sterile. So he went to school for a year in an astronaut suit. Eventually, I will sit up here and then I'll put them where they need to go. Appreciate it. All right, so I probably need to stop there. Again, that's a really interesting thing to look at. If you Google it, you'll be able to find it. We'll talk more about that later.